Hello dear students, welcome for today's session. So today's date 30th December. Okay, right. Now already we have seen a menstrual cycle. Let me revise at a glance. Now when you look at oogenesis, so the primary oocyte is present in the primary follicle, right? In the primary follicle. So in the primary follicle inside you can find now. So this is primary oocyte which is present in the primary follicle, and then it will develop into secondary follicle, and that will develop into tertiary follicle, and that will develop into Graafian follicle. Right? So primary follicles are made at the time of embryonic development. So see, when you say primary oocyte, already meiosis has been started and the cell has been arrested in prophase 1. Now these changes from primary follicle to secondary follicle and remaining changes will happen at the time of puberty due to action of follicle stimulating hormone. Then luteinizing hormone helps to break this Graafian follicle. Then the secondary oocyte is released, which generally we call as ovum. It is not functional ovum, it is a resultant cell of meiosis 1. By that you feel wonder, a cell is undergoing meiosis from embryonic period till puberty, and by the puberty time only meiosis 1 is completed, and it starts meiosis 2 and again it gets arrested in meiosis 2, and meiosis 2 is completed once the sperm cell enters, remember. So it's a very long journey which you need to note. Now this is the secondary oocyte. You need to know the structure, you may get questions here. Let us see the structure of secondary oocyte, means ovum. So usually it is a large cell compared to our body cells, and you can find the nucleus, and this is the plasma membrane. We can call it as oolemma, and you can find the cytoplasm, and some specialized granules are found. We call them as cortical granules. Cortical granules. So as usual, the cytoplasm, we call it as ooplasm. Then around the secondary oocyte, a new layer, protective layer is formed. This is what we call zona pellucida. This is zona pellucida. Just I am highlighting zona pellucida, right? Now at the time of rupture of the Graafian follicle, many of the follicular cells which are present around this primary oocyte, so they are still attached once it is broken down. I mean, so see, look at here. This is primary oocyte. Around this cortical cells or follicular cells are there. Now once meiosis 1 is completed, due to gonadotropins, due to action of gonadotropin, then at the time of rupture of the follicle, you can see a number of follicular cells around the secondary oocyte. It look like another protective layer, and this is called corona radiata. You got it? So once again, you look at here, at the time of embryonic development, primary follicles, so in each ovary you can find 2 millions of primary follicles, and the primary follicle inside you can find the primary oocyte, which has started meiosis 1, and it gets arrested in prophase 1. Then the further development happens only at the time of puberty under the influence of gonadotropins. So gonadotropins are produced like a follicle stimulating hormone and luteinizing hormone that will promote. So here also very interesting thing. So at the time of birth, if you look at each ovary, you can find 2 millions of primary follicles, and their number gradually decrease when a girl reaches to puberty. So at the time of puberty, if you look at each ovary, you can find 60,000 to 80,000 primary follicles. And so due to action of gonadotropins, every month only one primary follicle undergoes these changes. So you just remember, in each ovary how many are there? 60,000 to 80,000 primary follicles are there in each ovary. But here this gonadotropins influence only one primary follicle, either from left ovary or right ovary. 1 month right ovary, another month left ovary. Simultaneously they keep working, and in each menstrual cycle only one primary follicle gets converted into Graafian follicle, and due to effect of luteinizing hormone, Graafian follicle ruptures and the secondary oocyte is released. When you say structure of the ovum, you should understand we are looking at the structure of secondary oocyte. This is another confusion. So structure of the ovum. Here generally it is not functional ovum. Still meiosis is not completed. This is the resultant cell of meiosis 1, and technically we call it as secondary oocyte. What are the parts of secondary oocyte? First of all, look at the protective layers. Usually this is one of the animal cells. So plasma membrane is there. What do you call the plasma membrane? Oolemma. So and cytoplasm is present. What do you call? Ooplasm. And here there is another protective layer which is called zona pellucida. So here this zona pellucida is made up of glycoproteins. Remember, glycoproteins. That means a combination of amino acids or proteins and carbohydrates both together. We call glycoproteins. Note down, zona pellucida is made up of glycoproteins. Next, these follicular cells are also present around the zona pellucida, which we call corona radiata. So at the time of fertilization, you just remember this sperm cells has to clear up the corona radiata first, next zona pellucida, next plasma membrane which is called oolemma, then it has to enter inside the ovum. Right, once the sperm cell enters, meiosis 2 is going to complete. Right, so in the next video we will see the structure of this sperm cell.